Well, hello students, this is Mrs. Hanson, and we are engaging in our last lesson in our Chapter 20 discussion on carbohydrates. In your lecture guide, the note pack, we are about halfway down, about a quarter of the way down, I suppose, on page 12, where it says 20.5 disaccharides. We're going to just work together to complete the last few pages, knowing that the very last page you have a little reading assignment to do. So again, we're just practicing and getting a handle on what disaccharides, and in this journey we're just continuing that lesson and hooking two monosaccharides together to form what we call a disaccharide. See that prefix di, and of course we understand that that just is a prefix for two, so we see two monosaccharides are composing the disaccharide molecule. The linkage that holds these monosaccharides together is known as an acetyl linkage. Now remember, we were talking about a hemiacetal just in the previous lesson. A hemiacetal was the linkage used in a monomer, in a monosaccharide, when we changed it from the backbone, the long chain, into a cyclic structure. So if we had a 6-carbon chain, and remember how we had an aldehyde at the end and kind of going on to the end, how we reached up from an alcohol to create that cyclic structure and that linkage where we created a 6-membered ring, and that 6-member ring had a carbon 1 unit to the clockwise position, which would then reach up in an alpha or a beta position. This was known as a hemiacetal linkage. So in this journey, we're just going to take yet another monomer and create these 6-sided structures here. I'm just kind of drawing them in cyclics to go a little faster, but we end up making these 6-cycled rings connect with acetyl connections. The link there is known as an acetyl connection. So these disaccharides in this reaction creates what is known as a glycosidic linkage, just another name for what we refer to as an acetyl linkage. Very specifically in carbohydrates, they get a vocabulary word assigned to them as a glycosidic linkage. So in your notes, you have this drawn for you. I copy and pasted to remind ourselves, if this is a one structure like a monosaccharide, a hemiacetal linkage had this carbon at position number 1 attached to 2 oxygens. So the carbon, the anomer carbon, is attached to 2 oxygen in a hemiacetal linkage. Now compare this to a glycosidic linkage, where the anomer carbon in molecule 1 is positioned to attach to yet another cyclic structure from its position in number 4. And we'll practice this in our drawing. A glycosidic linkage is between 2 monosaccharides. And I want you to notice how we go about connecting one monosaccharide with another monosaccharide. To do so is a dehydration process, that we have an H and an OH being removed to create the alcohol, this linkage here from the alcohol to create the glycosidic linkage. Very specifically, we have a carbon attached to another oxygen that leads to another oxygen that then is attached to another cyclic ring. And very specifically, you'll see that in a 1,4 glycosidic linkage. Carbon number 1 from the first monosaccharide is going to attach to carbon number 4 in the second saccharide. And so notice that C1 from ring 1 bonds to C4 from the second ring to create a glycosidic linkage. And to do so, we dehydrate, creating, minus HOH, creating the linkage there between the 2 sugars. Now this glycosidic linkage joining the 2 rings can either be alpha, which remember is pointing down, or it can be beta, which we've learned means that it's pointing up. If the bond is an alpha, notice that from the carbon position 1 in this ring, notice that that's carbon 1, and in this ring, this position is carbon 4. Both are pointing down, and so therefore we have an alpha glycosidic linkage. The glycoside bond is pointing down, indicating it's an alpha glycosidic linkage. If you see in the opposite trend that we have pointing up, this original group here from carbon number 1 is pointing up, we know that it's now called a beta glycosidic linkage. Carbon 1 from ring 1 bonds to carbon 4 from ring 2, and if it's pointing up, we call that a beta linkage. So a lot of opportunity to just apply from monosaccharides to the disaccharides. Here's a practice. It just asks us to locate the glycosidic linkage in maltose. Notice here, this is a disaccharide. It's composed of 2 monosaccharides. The glycosidic linkage is from the alpha, the anomer carbon here, leading all the way to carbon 4 on the second ring. Carbon 1 from the first ring attaches to carbon 4 from the ring in the second monosaccharide. Since this is pointing down, we understand pointing down means that it's an alpha glycosidic linkage. When we number, we always start with carbon 1 being the anomer carbon. That's the carbon located 1 position clockwise from the oxygen that's part of the membered ring. And so numbering all of those shows this 6-carbon glucose, starting at the anomer carbon, which is always 1 carbon clockwise to the starting oxygen in the ring. We can see that this is also a 6-membered glucose, and we have a 1,4 alpha glycosidic linkage in the sugar called maltose, a disaccharide. And this is just exactly what we drew. Here we have our carbon right here, 1 clockwise from the oxygen, creating a 6-membered ring with oxygen as part of that 6-member, always right up in the right-hand column. And when we number those, the 6th carbon is up above the plane. So that's just an opportunity to compare your picture with mine right here. We've had an opportunity, just backing up, to create an alpha glycosidic linkage. You drew that on the bottom of page 12. You then were asked to draw a beta glycosidic linkage, and so you do that on the right-hand box on page 12. And we're deciding that based on the position of the glycosidic linkage in maltose, that we labeled that as an alpha glycosidic linkage, and you were asked to number each of those carbons. If you're ready for another, let's try the next. Here's an opportunity to practice with this question. Locate the glycosidic linkage in cellobiose and then number the carbon atoms in both rings. So the glycosidic linkage, locate the carbon that's 1 unit clockwise in ring number 1 from the oxygen in the ring. Carbon number 1 in the first ring is always clockwise to the oxygen, and I'll number around, making carbon 6 1 unit above the cyclic structure. Again, in this next ring, 1 unit clockwise to the oxygen that's in the ring is always carbon 1, and I'll number around, making this also a 6-carbon ring. The carbon in the circle 1 connects to the carbon in number 4, and the glycosidic linkage are all of those atoms that bond the 2 cyclic structures together. Since this is pointing up, we would label this as a beta glycosidic linkage. There. And there we go. Good work. This is coming along. So if we have, in this particular picture, just to kind of add emphasis to what we just drew, this is your check of the picture you just drew. We numbered the glycosidic linkage as the bonds between the first carbon, position 1 on the cyclic structure, and that glycosidic linkage is attached attaching the two cyclic structures. We labeled this as beta since it is pointing up. An opportunity to check your picture against this diagram here. Well, another topic related to disaccharides is the ability to cleave the bonds in the CO linkage. The glycosidic linkage can be cleaved, which means to break apart through the process of adding water back in. So upon adding water, we're going to go back and create the initial alcohols, locate the glycosidic linkage. Remember, at carbon number 1, it leads out to that oxygen. This is the site then that is going to get cleaved, which means to be broken, when we add water to break apart the two monomers back into their building blocks. Notice this is alpha, so it's pointing down, which means the positions here will be pointing down. So all I have to do is to redraw each monomer, but break the position here and reestablish the alcohols. Since the linkage is alpha, both alcohols are drawn down. And you might want to pause to just catch up your drawing if need to. If we have a beta position, what monosaccharides would be formed if I hydrolysize this disaccharide with water? Locate the glycosidic linkage, and that's the bond that is going to break and reestablish the alcohols. So hydrolysis means to add water across the bond. You're going to break the glycosidic linkage and form alcohols in both positions. And then just take a note if it's alpha or beta. Notice this is pointing up, so at carbon number 1, as you continue to draw that, let me make that neater. As you continue to draw carbon 1 right here, the alcohol will be pointing up because it's a beta alcohol. And then, of course, everything else will go in just as it appears in the original drawing. And, of course, here we have that 6 carbon, CH2OH. So this is from your ring 1, noting that the beta position is showing that this was a pointing up glycosidic linkage. And then as we redraw that second monomer, remember it's carbon number 4. At carbon 4, what we end up seeing is that OH is pointing down so that it actually had a site of attachment. So at carbon 4, we point down so that when I break this glycosidic linkage, one on top, one on bottom, and we'll see that pattern then continue. Here we had HOH at position 1, position 2, at position 3, the alcohol's on top, and of course at position 5, we reestablish the CH2OH link. So we just broke apart through hydrolysis, adding water across the bond, and just paying attention if we need to draw it up or down based on an alpha or a beta configuration. So again, just an opportunity to look and see that those are pointing up and down based on the beta configuration of the original disaccharide. We'll turn our page. Top of page 14 talks about polysaccharides. It says lactose intolerance. Let me see. Yeah, polysaccharides. This is just kind of finishing up on that last one. On the bottom of page 13, I guess I forgot to just mention there that when we look at this lactose intolerance, there's just an opportunity to bring in a little bit of health and medicine ideas. Nothing to write down on this slide. We have a galactose and a glycosidic linkage here with a glucose attached. And this is something you did in the lab where you broke apart the glycosidic linkage in lactose and recovered glucose and galactose. So nothing to write there, just kind of a nice to know about lactose intolerance. something that relates to some people who have lacked the enzyme to digest this lactose sugar. And now moving forward to 20.6, the polysaccharides. They contain 3 or more monosaccharides joined together. And so again, just repeating units with glycosidic bonds. So same glycosidic bonds. And the 3 most common that we see are cellulose, starch, and glycogen. Here we notice that we have a beta glycosidic linkage because it's pointing up between 2 monomers. And when you see this little continuation on both sides, it just means that this pattern keeps repeating over and over and over. So again, notice the little squiggles, the patterns are repeating over and over and over. And this is emphasizing that this is an alpha glycosidic linkage because it's pointing down in that particular structure. So starch and glycogen, their repeating structure is an alpha position, and cellulose has a beta position. Polysaccharides contain at least 3, must be at least 3, otherwise it'd be considered a disaccharide. And they're joined together by glycosidic linkages, again through the 1,4 alpha or beta. If it's cellulose, we saw it beta. And if it was starch or glycogen, we saw it in an alpha position. And just a character or quality of cellulose, we find that in cell walls of plants where it gives support and rigidity to wood, plant stems, and grass. Cellulose is a very fibrous substance. For instance, found in celery, the very fibrous, woody pulp that celery can degrade to. And humans are not able to digest that particular type of substance, the cellulose molecule, because we lack the enzyme to hydrolyze. We can't break down those glycosidic linkages, and therefore it comes out in the same fashion as it went in. Which is fine because it actually adds a little consistency to our solid wastes. And that's its purpose, is to increase your fiber and you have a more solid waste product. So again, here's cellulose. You notice that each time the glycosidic linkage is pointing up, shown in blue. So this is a beta for cellulose. Starch is a polymer made of repeating glucose units joined by an alpha glycosidic linkage. That means they'll be pointing down. So the glycosidic linkage of starch in a repeating pattern. This is a molecule of amylose. So the first main type of starch is amylose, and we find that in corn, rice, wheat, and potatoes, some of the main food sources for starches, especially potatoes. But here you notice that it is an alpha glycosidic linkage. And the second main type of starch is amylopectin. Amylopectin has alpha linkages between the monosaccharides, and then we actually see a linkage that ties together branches along one chain to the next. So it's a little more complex molecule than that of the amylose of the previous slide. Amylose was just a straight chain, but here we're seeing the amylopectin is more complex because one chain can attach to another chain through these side branches right here. So one carbon can reach up and form an oxygen, so it's definitely a more complex molecule. Those amylose contain branches that are 1,4 alpha glycidic linkages, as well as amylopectin containing 1,6 alpha glycidic linkages to create a more complex chain where more connections create branched molecules. Both of these starches are digested through the enzyme called amylase. And friends, that was the rest of our chapter. The rest is really just up to reading. You might have been able to fill in the notes just from reading, but this is just a valuable lecture to kind of just check in and make sure you heard from my voice all the things that I thought were important to record. So use your text to complete the last few pages as a reading assignment, and I'm looking forward to receiving your completed lecture guide in the Chapter 20 Dropbox before the end of today. Have a good day.